Thank you for joining Continuous Learning with Bioguard. It is my pleasure to introduce our webinar speaker, Dr. Chen Xilin. Dr. Lin got her DVN degree from National Taiwan University and her PhD from the College of Biological Science and Technology, National Jiao University. She's a professor in the Department of Veterinary Medicine and Director of the Zoonosis Research Center at National Taiwan University. In addition, she is a former director of the Animal Disease Diagnostic Center at National Taiwan University. Her specialties include veterinary clinical microbiology, clinical pathology, and animal cancer biology and therapeutic development. This lecture today will explore the clinical application of bacterial culture and antimicrobial susceptibility testing to guide responsible antimicrobial use. It covers proper sample handling, key diagnostic methods, and interpretation of the resistant data with clinical case. We are thrilled to have Dr. Lin as our speaker for this webinar. Without further ado, please join me in welcoming Dr. Lin.
Hi, everyone. Thank you for the introduction from Dr. Luo. And it's my pleasure to join the webinar tonight. So first, I will share the slides. Okay. So we will know a little bit about the clinical veterinary bacterial culture and the antimicrobial susceptibility in clinical sites. So I will start the lecture. So I will share some of the knowledge as well as the experiences. So any questions from you are welcome to we can be discussed further. So again, good evening, everyone. I'd like to begin today by emphasizing the role of culture and susceptibility testing in modern veterinary practice. Too often, antibiotics are prescribed and emphatically based on clinical impression rather than laboratory evidence. While this approach may provide long-term relief, it can also drive the development of antimicrobial resistance and make future infections harder to treat. So when we rely solely on empirical antibiotic use, we face serious consequences. Resistant infections increase treatment failures, prolong recovery, and in some cases, lead to systemic complications or even mortality. Moreover, resistant bacteria in dogs and cats can be transmitted within households, creating a One Health challenge that lacks animal, human, and environmental health. But I need to focus on that. I mentioned in this point, it's not just on animal doctors, but we also need the cooperation from human doctors, because the proper usage of antibiotics actually is a very global, important global issue right now from the aspect of One Health. So that doesn't mean that only animal doctors we use the antibiotics improperly, but also actually we also see some cases in human sites as well. So in small animal practice, culture and susceptibility testing is especially relevant in several common indications. For example, Pyoderma. In Pyoderma cases where Staphylococcus intermediates can rapidly develop resistance to first-line drugs. And another example indications that will often for the animal doctor to use antibiotics is otitis externa and media. And in these cases where mixed infections, different kind of bacterial infections could occur and may require tailored therapy. And sometimes there's the other common cases will be urinary tract infection, and this kind of urinary tract infection often involving multi-drug resistance E. coli. And less common case we see in clinical would be respiratory infections. And these respiratory system infections where empirical treatment may fail if the pathogen is resistant. So by adopting proper bacteria culture and susceptibility testing more constantly, we not only improve outcome for our patients, but also contribute to global efforts in antimicrobial stewardship. The data we collect helps build local antibiograms, guiding practitioners toward more effective first-line choices while preserving critically important drugs for cases where they are truly needed. Okay. So the next I will share some data we've obtained from our animal hospital in our university. So in our university, this study actually is already published, and we collect many samples from the animal cases, and we perform some. We try to investigate the dominant bacteria infection as well as the potential antibiotic resistance phenomena. So actually in Taiwan, the studies conducted in our animal hospital and has shown that the prevalence of ESBL, what ESBL means, extended spectrum beta-lactamase. So this kind of enzyme, because lactamase is some kind of enzyme possessed by the bacteria. Once the bacteria, they have this kind of enzyme, it means they have significant resistance to antibiotics. So in Taiwan, our study have shown that the prevalence of ESBL-producing organisms, the ESBL harvesting bacteria in canine and feline clinical isolates, is around 12 to 13%. Among all the cases that within this investigation. While the number may appear modest at first glance, right? But it reflects a substantial risk, particularly because many of these bacteria are multi-drug resistant. So this is what we want in Taiwan situation, that although only around maybe 12 and 13%, however, because these bacteria are multi-drug resistant, that means from these clinical cases, the veterinarians, we face the problem that we do not have effective drug. We do not have effective antibiotics to treat these cases. So the most common species we encounter include E. coli. Yeah, so that's why we said that this is the many cases we can find the E. coli and they possess the multi-drug resistance. And so next to E. coli will be Klebsiella, Enterobacter, Acinetobacter, and Serratia. So these are organisms that cause common infections such as urinary tract infections, wound infections, and the severe cases would be sepsis. So once they acquire ESBL genes, treatment options become limited, often requiring the use of a higher titer and tighter or last resort antimicrobials. So from a genetic perspective, the TEM, this TEM, and the CTXM families are the predominant resistance determinant, with the CTXM1 group being the most frequently identified in common animals. These enzymes are highly efficient at hydrolyzing third-generation antibiotics, including cephalosporins, making drugs like cefotaxime and cefotaxime ineffective. So this is very, actually it's the higher generation cephalosporins. However, if the bacteria harvest this kind of genes, that makes this bacteria, they are resistant to this kind of antibiotics. So another important point is the clonal spread of resistant strains. In Taiwan, for example, the major E. coli sequence type identified is ST457. The presence of the defined sequence type suggests that these resistant bacteria are not arising randomly, but are spreading within populations, potentially even across species barriers. For us as clinicians, the key message is when faced with infections caused by ESBL-producing organisms, empirical use of cephalosporins is unlikely to succeed. Culture and susceptibility testing are essential, and we need to reserve higher priority antimicrobials for cases where no other effective options remain. This is where antimicrobial stewardship becomes not just a recommendation, but a professional responsibility. So now let's turn our attention on E. coli ST131, one of the most clinically significant clones of E. coli worldwide. In Taiwan, actually ST131 has been identified as the second most common ESBL-producing clone, accounting for roughly 15.4% of isolates in dogs and cats. This is particularly concerning because ST131 is well recognized in human medicine as a major driver of multi-drug resistance, especially resistance to fluoroquinones and third-generation cephalosporins. What makes ST131 especially important in its dual presence in both pets and humans, this highlights the possibility of interspecies transmission. In other words, the same resistant bacterial lineage can be shared within a household, moving between people and their companion animals. So this has been demonstrated in several international studies, and the findings in Taiwan reinforce that this is not just a theoretical risk, but a real-world concern. So when we look at antimicrobial resistance trends in companion animals, one of the most challenges we face in Taiwan is the lack of comprehensive longitudinal surveillance data. Unlike some regions where multi-year monitoring networks are already in place, our picture is still fragmented, often relying in single institution or short-term studies. such as the study that I just shared with you in the previous slides. So to place this in context, let's consider findings from other areas, such as from Northeast China. Between 2012 and 2013, the prevalence of multi-drug-resistant bacteria in pet dogs was reported at nearly 77%. So by 2021, this number had dropped to about 62%. That decline may reflect improvements in antimicrobial stewardship, regulation, or clinical practices. However, the fact that more than half of isolate are still multi-drug resistant illustrate the scale of the problem. So interestingly, while pet dogs showed this decline, working dogs demonstrated the opposite trend, with resistance rates actually increasing. This raises an important question about differences in management, antibiotic exposure, and environmental pressure. So we also see that the cats tend to exhibit higher levels of antimicrobial resistance than dogs. This may be due to differences in physiology, husbandry, or diagnostic practices, but it highlights that the species-specific considerations are important when developing treatment guidelines. So on a more positive note, both colistin and carbamazepines remain largely effective, with resistance reported in less than 2% of cases. These drugs are critical last-line therapies in both human and veterinary medicines. While their continued effectiveness is encouraging, it also reinforced the need for strict stewardship and the advance of unnecessary use of antibiotics. So we just shared the information gathered from some specific nations. So the trend will be different from other area around the world. However, I think try to use the antibiotic properly and before treatment, if we can perform enough tests to know what kind of antibiotics is the proper drug that we should use is the most important issue that we should do. So actually, I think, just as I mentioned, to know what kind of bacteria in the infection cases and what kind of antibiotics we should use as the most important thing to prevent from the increasing of antibiotic resistance. But the first step that we need to know is about how to perform the proper sample collection and handling. So getting reliable culture and susceptibility results always begins with how we collect and handle the sample. Even the best laboratory cannot overcome a poorly collected specimen. So first, we need to use aseptic techniques. This means minimizing contamination right from the start. For instance, in urinary tract infection, a cystosantinase sample provides a much more accurate picture than a free catch sample, right? Which is often contaminated because the free catch samples is often contaminated by the lower urinary tract or environment. So similarly, when swabbing ears or skin, preparing the site properly reduces the chance of recovering only surface flora instead of the true pathogens. So this stage actually is really important, the right way to use the aseptic techniques. So second is we need to notice is about the contamination prevention, because this step is absolutely critical. A contaminated sample can lead to false results, sometimes showing resistant bacteria that are not actually causing the infection, or sometimes masking the real pathogen. This can easily lead us to select the wrong antibiotic, which harms the patient and drives antimicrobial resistance. So again, the well-prepared aseptic techniques and to prevent the possible contamination as the key steps for the sample collecting and handling. And third, we must pay close attention to transport considerations. Think about the medium you use to store the sample, the temperature during transport, and how quickly the sample reaches the laboratory. For example, respiratory samples from our nose collected from nose, for example, or class from the trachea. This kind of respiratory samples left too long at room temperature may lose viability of fastidious organisms. Because this kind of organisms, for the fastidious organisms, because they don't like oxygen. So if this kind of sample contains with this kind of bacteria, they may, if they stay at the room temperature for a long time, they got a lot of chance to expose to the air, exposed to the oxygen. So finally will lead to the death of this kind of bacteria. So finally, we may not have the good result of the bacterial culture. So, so such as I mentioned, respiratory samples, if we left them too long at the low room temperature, may lose their viability and leading to the false negative results. So ideally, samples should be processed within a few hours. So it's very important within a few hours. So finally, don't forget communication with the diagnostic laboratories, providing details such as the suspected sites of infection, the species, and any prior antibiotic treatments helps the lab staff choose the most appropriate culture methods and ensures the results are interpreted in the right clinical context. So I always think it's the really important step. Right now, I think each central lab or each diagnostic labs, including us, we design the very well prepared documents for the clinical doctors to write down all the messages, all the possible messages about the case. So it helps both the diagnostic lab staff as well as the veterinarians. We can connect the good communications, and so it can help both of us to interpret the result in a good way. So in summary, accurate diagnostic results are not just a laboratory responsibility. They start with us by applying correct techniques, preventing contamination, handling sample properly, and collaborating with the lab staffs, the diagnostic labs that can help us greatly improve diagnostic accuracy and support antimicrobial stewardship in our daily practice. So now we can move on about the how to collect the sample properly. So this is the very important steps because even the best lab cannot give us accurate results if the sample is poor. So yeah, so here we just provide some common lesions that we will need to do some kind of a bacterial culture. We will need to collect the sample from basically from these main four lesions. So start with the skin. So always clean the for the skin sample collection. Always clean the area with alcohol first, right? I think it's a basic step that we will perform. And then collect the sample from the active age of the lesion. And so if the skin, you got some lesion on the skin, so just disinfection with alcohol first, and then collect the sample from the active age of the lesion. This is where the bacteria are alive and the most representative. So if we only swab the center of the old lesion, we may just get debris or contaminants instead of the real pathogen. So it's very important. The key issue that we need to, when we doing the sample collection, we collect the sample from the active age of the lesion, not the center of the lesion. Because during the center, there's always a lot of nucleus host cells and a lot of dead bodies of bacteria. So that's actually sometimes even the veterinarians, they told us that, we've already gave you a lot of samples. So how come the results, we still got no bacteria, we still got no antibiotics usage results? That's because the first step is very important. You need to get the uncorrected site to collect the samples. Yeah, this is for the skin way. So for the ears, if we need to collect a possible infection, we will diagnose the samples from the ear. So when you collect an ear swab, we use swab, right? So when we collect an ear swab, try not to touch the canal walls. If the swab touches the surface on the way, you can easily pick up commensal bacteria. Commensal bacteria means the bacteria that actually is coexist with the host. So this kind of commensal bacteria, they are not the main pathogens to cause the infection. So if we try to get the ear swab, just don't try your best not to touch the canal walls. Yeah, it's very important. So because if we pick up only commensal bacteria, and the culture won't reflect the actual infection. So for the urinary tract, the best option, I think everybody right now knows about the cystocytosis. Yes, it's more invasive actually than free catch urine, but it gives us a sterile sample directly from the bladder. Free catch samples are often contaminated and can mislead us into prescribing the wrong antibiotics because this may contaminate it from the lower urinary tracts, from the environmental. So yeah, this free catch samples, we may get a lot of bacterial results. However, this bacteria may be not the main pathogens to result in the bladder infection. So yes, this is the thing we need to pay attention. Also the respiratory tract. If you suspect a lower airway infection, a bronchial lavage, or a protected brushing, it's far better than a simple oral swab, right? Because if you find this is the lower airway infection, not the upper airway, so we need the bronchial lavage may need to perform. So these techniques minimize contamination from the mouth and give us a much cleaner picture of what's happening in the lungs. So the key messages here is good culture actually results from the good sampling. If we take care at these steps, we save time, avoid mistakes, and provide much better care of our patients. So then we move on to the culture result interpretation. So once we have culture results, the next step is interpretation. And this is not always straightforward. First, we need to think about pathogens versus contaminants. We've mentioned that there are some possible contamination could be occurred during sampling, right? So not every bacteria that grows in culture is actually causing disease. So for example, in ear swabs or skin samples, we may recover commensal or so-called normal flora or environmental contaminants. So the key is to combine the culture results with the clinical size. So right, we can. I think we cannot just believe one culture reports and we say and we defined the case with what kind of infection. So we need to combine with the clinical side, try to help us to get the comprehensive pictures of this case. So asking, does the organism really explain the infection we're seeing, or is it just alone for the right? Yes. So this is the clinical side that actually the culture report may give us different kind of bacteria. So which bacteria is the main pathogen? We need to ask ourselves by the different the clinical site and maybe the other lab tests that you've already done, and try to differentiate which one is the most possible bacteria result in this kind of infection. So we need to see, when we, during our interpretate the culture results is about the growth patterns. So a pure growth of a single organism, especially if it matches the clinical size, is usually significant. But the mixed growth, for example, in wounds or respiratory samples, can be harder to interpret. In some cases, it reflects a true polymicrobial infection, but in others, it may simply be contamination from surrounding flora. So knowing what counts as normal flora in each body site is essential to making the right call. So finally, let's talk about the negative cultures. I think I think maybe sometimes you will get this kind of reports, right? The reports will say, oh, the sample, we work very hard to take actually with no, they found no bacteria grow up. That's the negative culture. So the negative result doesn't always mean there is no infection, right? It could be a false negative, perhaps due to prior antibiotic treatment, poor sampling, or transport issue, or cultural issue, I need to say. On the other hand, it may also mean the problem is not bacterial at all, but if it's not bacterial, maybe we should consider other pathogen infections such as fungal, virus, or maybe just inflammation. So in those cases, further diagnostics are needed. So the big message is interpreting culture results requires both microbiology knowledge and clinical judgment. We can't just look at the lab report in isolation. We have to integrate it with the patient's history, signs, and response to the treatment. So, okay. So that's so let's move on to an overview of the susceptibility testing. So if we got some possible culture regards some bacteria already we have, the next stage actually, this is for the conventional way. We need to do bacteria culture, and the next step would be the susceptibility, antibiotic susceptibility testing. So how we actually determine which antibiotics are likely to work. The two main laboratory methods are disk diffusion and MIC testing. This diffusion is simple and widely used. So antibiotic discs are placed on an agar plate, such like this. Okay, on the agar plate agar plate seeded with bacteria. So these agar plates are already with the specific bacteria that we cultured, and they grow up on the agar. Then we plate and we measure, and then we put the different kind of antibiotic disc, and we measure the zone of inhibition. So this is disk diffusion. And for the MIC testing, our minimum inhibitory concentration is more precise. It gives us the lowest concentration of an antibiotics that prevents visible growth. Both methods provide valuable information, but MIC testing is often more useful in complicated or resistant cases. Yeah. And to interpret the result, we rely on CLSI standard. The CLSI standard means the Clinical and the Laboratory Standard Institute. They classify result as S for susceptible, and the I for intermediate, and the R for resistant. So understanding this category is essential. So you can read their report is S, I, or R, that means. So for example, but I think maybe most of us will be confused about what is the intermediate, right? Intermediate does not always mean the drug is useless. It may still be effective, but at higher doses or in certain body sites, such as the urinary tract, where drug concentrations are higher. Of course, no test is perfect, so we need to recognize their limitations. Okay, so finally, a special note on multi-drug resistance and the ESBLs. For this kind of ESBL organisms, they can appear susceptible in standard testing, but behave differently in vivo, especially with cephalosporin. So this makes correct detection and sorry. To close this. Okay. So I just mentioned about this kind of ESBL bacteria. They can in the culture way, they show the susceptibility. However, they may not actually in vivo. In vivo, that means in clinical site. Actually, they can. So it's very special that for this ESBL organism, they can appear susceptible in standard testing, but behave different in vivo. That means on the report, you can see they are sensitive to specific drugs. For example, they are sensitive to cephalosporin. However, for the clinical, if you treat the case with cephalosporin, and it still has no effects. So this makes correct detection and the cautious interpretation extremely important. So susceptibility testing gives us a scientific foundation, but as clinicians, it's our job to integrate those results with the patient's history and site of infection and overall health status. And I think the most important is your experiences. So yeah, we've mentioned a lot about the antibiotic susceptibility testing, and we think when we should do this before the start of any treatment with antibiotic drugs. It's very important. So we should not to perform try and error thing, especially on the usage of the antibiotics, right? So before the treatment, we should to do this AST and antibiotic susceptibility testing. And also during the treatment, if we want to monitor the effectiveness of treatment, we can perform AST. And the third condition would be when the infection is not responding to the treatment, that means maybe something happened. Maybe because of the progression of the disease, maybe we need to redo the AST testing. So as we have gone through about the AST testing, the first we need to grow up to do we need to wait for the grow up of the bacteria. This usually will cost us 1 to 2 days, 24 to 48 hours. And then we can have this bacterial colonies, then we can perform the AST. Yes. So this is what we usually do, and it will cost a lot of time. So actually right now, we have, this basically for the lab, we performed an AST to give us about the result. And for right now in AST testing in both veterinary and human medicine, and there are so many ways for us to get the AST results. So this is the summary of the methods that right now we've used. So first, we have the phenotypic methods. This includes the dilution, disk diffusion, and gradient test. So when I say this, they are time consuming and usually taking 24 to 48 hours and sometimes longer to get the results. And second, we can use the molecule-based methods such as PCR and the qPCR. And finally, if we really want to know the precise and the diagram, the overall picture of the infection, we can use mass spectroscopy, especially MALDI-TOF. Right now, it's becoming actually it's already the regular methods in the human medicine, but for the animal medicine, it's becoming popular. So it identifies bacteria rapidly and can be combined with antibiotic incubation for resistance profiling with a few hours. But the only thing we need to consider is about right now it's popular in human medicine, but not in vet because it's very cost a lot. We have now that we need to do for the regular testing, We can do the diffusion, we can do MS testing to give us a more precise about the minimum inhibitory concentration, and we need Okay, I'm sorry. So is anything wrong? Okay, so what I mentioned is about, yes, we need to do about this. So for the conventional antibiotic susceptibility testing, it's labor intensive, and we have the long preparation and incubation time. And also we suffer from the high risk of cross-contamination. And it's expensive and need a high power supply and prone to human error in analysis. So right now, I think if we have other options about the test, why not if we just go a step further? So this is right now, there are several kind of devices to help us performing the we do not waste a large, we do not need to waste much time, and we can easily to get which kind is the proper antibiotics we can use. I use this microfluid device. So for example, this is the AST machine provided by Bioguard. And this is the, you can realize that the machine, the figure of the machine actually is not big. You can just put in your clinician office. And actually, it just as I said, this is microfluid device. So you can put your sample and following the guidance of the reagent packages, and then you can easily get the result. Yeah, you can collect the sample, and the sample can use this kind of device will be such as the urine, the fecal, skin, ear swab, wind swab, tracheal swab. This kind of samples could use this kind of device. And finally, this kind of microfluid-based device could give you the results with different kind. They have different kind of antibiotic portfolio, such as we show here: panel 1, panel 2, panel 3. Or you can design your own panel. If your animal clinics, they prefer some kind of antibiotic panels because of the case usually exposure. So it's also could be tailor-made. So it's the result. So you only put your sample with the machine, and after 6 hours, they can give you the results. And you can, as we can see, that the result is about, this case sensitive to amoxicillin and resistant to cefazolin, such as that. So it's based on the COSRI guideline that you can just read the S and R and I to find the specific drug that you can use. So actually, it's you can use either way. However, finally, you will get the result. So this is what I mentioned. You get the report, you need to do some correlation from your clinical findings, right? And then during the treatment, if you want to follow up the treatment is effective or no. So during the treatment, you can also do another round of susceptibility test, and it could help us to adjust or to manage our therapy. So, for example, there's some kind of case. Yes, this is not the real dog in this case. It's just, for example, picture. But I think it's cute because yes, this Labrador, and Labrador always cute, and they intend to get bigger, the bigger guy, right? Yeah, because I think they have the genetic. Right now, some study have found they have the genes that make all the Labradors, they like to eat everything. They are food lovers, think. Okay, so this case, actually, they have the UTI UTI infection in already in 6 months. So previous empirical treatment with amoxicillin, the amoxicillin and clavulanic. This is the treatment history. So again, for this UTI infection, they get the urine sample and to do the bacterial culture and antibiotic test. Finally, they found it's the E. coli resistant. It's the E. coli infection. However, this E. coli is resistant to amoxicillin, but susceptible to trimethoprim, sulfur, and enrofloxacin. So it's I think it's a very simple case that we've already seen many times. And so finally they try to do some clinical decisions. Yes, based on the antibiotic test results. They choose the proper antibiotics and extend treatment course and follow up culture. So again, they do another round of culture right now. So finally, the case is okay and discharged. So I think it's a very simple case. However, it showed us the standard protocol that we need to concern about the antibiotic AST test. So before the treatment, during the treatment, it's very important. So this is for the dogs and cats, I think we've already deal with a lot of cases. So how about the exotic animals? Because exotic animals actually they include a large and different species, right? So for the unique challenges, if we concern about the bacterial culture or ST test of exotic animals, the challenge would be, for example, small sample volume, limited drug safety data, right? We have a lot of safety data of different antibiotics for the companion animal, for dogs and cats, but this is almost few for the exotic companion animals, right? So most of the case, I think, will be off-label use. So I just listed the common pathogens in exotic species. For example, in rabbits, we can see Pasteurella matosida, Staphylococcus, and also in other specific species. So there is some swab tips that we need to take care of, but later I will have the slides the slides will share to you so we can see the tips here. Yeah, so it's the again, I think the for the exotic species, we need to concern it's about the species-specific toxicities, because many antibiotics that are safe in dogs and cats may be toxic in exotic species. So it's the very important issue. So that's very for the owner education and husbandry reaction actually is important for the exotic species. Yeah, this. So actually use culture results to guide target therapy and try to select narrow spectrum antibiotics when possible. It's very important. And also, I think it's also important to document the decision-making in medical records, because it helps us to understand the history of what kind of drugs, what kind of antibiotics we've used. So finally, I want to share with all of you about the culture and the susceptibility testing will enhance precision and reduce resistant risk. And always interpret results in clinical context, not just in isolation. We cannot just count on isolation, and involve diagnostic labs early for optimal test selection and guidance. I think everybody knows that they promote responsible antibiotic use in both small animals and exotics is, I think it's everyone's responsibility. So thank you. Thank you. And since my talks end here, now I will sorry. I can see the comments and questions and from the audience. Advice can be given very beginner. Yes, and one question from the it's a very good question that because of I think yes, actually I think even in Taiwan we don't have many clinicians, many clinic small animal hospitals. We don't have our own culture system, yes, because it's always as I mentioned, it's time consuming and labor intensive, right? So we don't have this kind of our own culture system. So yes, we can just try some specific antibiotics maybe. But just as I mentioned in this slides, if we have our own small machine, this I've mentioned the microfluid based an AST machine that you can only need to spend maybe just 6 hours, and you can get the result of, what kind of antibiotics is sensitive. The bacteria is sensitive to what specific antibiotics. So if you got this kind of machine, actually you don't need to spend a lot of time. You don't need another person. You just put your sample into the culture, into the micro fluid-based machine, and then you can get the result after 6 hours, and the result will tell you the proper antibiotic you should use. Yeah, it's very interesting that the working dogs and the cats actually they are sure higher antimicrobial resistance. It's a very it's a very important and interesting issue. And right now, the study have just showed the epidemiology data, and they try to explain maybe due to the environment they contact, they are not they are not like the dogs and cats in the family. They are well take care, right? So the environmental may be one of the factors, as well as the working condition, many different kind. I think it's very complicated issue. So right now, I think the specific reason are still finding, but I'm sure that this is the complicated one, not just because of the specific pathogenesis of reasons, I think. Okay, so still some questions. What disk? Okay, if we're dealing if we are dealing about the goats with mastitis, actually it's it's very interesting. It's the farm, because goats, I think, is the farm animals, and again because of the mastitis, so yes, you can get the proper sample, but yeah, mastitis, yeah. For the mastitis, because maybe the sample would be the milk. Would be, probably would be the milk thing. So, what is typically applied? What disk concentration? Yeah, it's at for the mastitis and goats thing. I think I can have I can look up some possible findings for you, and later I will, if I find anything new, I will reply to the Dr. Luo, and I hope you can. Get the data I've collected for you. Okay. So thank you for your thank you for your joining us, and I will share the slide later. So thank you.
Thank you, Dr. Lin, for the excellent lecture. And thank everyone for joining us today. We'll see you again next time. Thank you.
Thank you.